Hello, everyone, and welcome to today's live broadcast, Metabolites, Germs and People, Eavesdropping on Human Microbial Communities. It is presented by Katrine Whiteson, PhD, Assistant Professor of Molecular Biology and Biochemistry and Pediatrics at the University of California, Irvine. I am Judy O'Rourke of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. The speaker will answer your questions via email. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. I want to let everyone know that today's webcast will be available for on-demand viewing through July 12th, 2018. You will receive an email from LabRoots letting you know when the webcast will be available for replay. Please share the announcement with your colleagues who may have missed today's event. Please join me in welcoming Dr. Whiteson. I will now turn the presentation over to her.
Thank you very much for the introduction, Judy. I'm happy to get to present my work today. Thank you for the opportunity. I'm speaking to you here from UC Irvine in Southern California. So we're between LA and San Diego. And I started a lab here at UC Irvine 3 years ago, focused on the impact of human microbial community colonization on human health. And as you can see in my picture of my group here, every year we paint a mural, and this one is called The Phages of Orange County, because we joke that there's more phage than housewives in Orange County. And so today I'm going to tell you about a few of the projects in my lab and also just give you a general introduction to the impact of the microbiome on health. And then I just definitely want to make sure that I acknowledge my group members. As I mentioned, I started here 3 years ago, and these are some of my graduate students and undergraduate students, and also a bioinformatician with a PhD who have been helping with all of these projects that I'm going to present today. So whether you're a human or a fly or really any life form, you're colonized by microbes. The microbes got here before we did. They've colonized the Earth for 3.8 billion years. And each of us have a unique community that stays with us for our lives. They assemble in the early parts of our lives and actually stay quite unique and distinct between people. If you look here, I'm showing you the host-associated microbiota and the density of the microbes in different sample types. So for example, you'll see that human colon samples and human dental plaque samples are some of the densest microbial communities on the planet, even more so than some famous environments that you may have heard of, like soil and ocean sediments. Really, colon and dental plaque samples are very densely colonized, which means that we have an enormous number of bacterial cells, which are really part of our bodies in a way. And each of those bacterial cells are infected by another order of magnitude worth of viruses. One of the biggest consequences of this is that we have a huge number of bacterial genes with very diverse metabolism that outnumber our human genes by many orders of magnitude. And so what that means also is that most of the genetic differences between any two people are actually encoded in our microbial communities. So you and the person sitting next to you right now have a lot of different metabolic capacities that are brought to you by the microbes, especially the ones in your gut. And in the last 10 years, especially since DNA sequencing has become less expensive, we've really come to understand that disease can arise from a disturbed microbial community. So if there were any two things that I could ask you to remember from this presentation, they would be these two things. That first of all, microbes are found in every Earth environment. That means that nothing is sterile. Even if your medical textbooks are talking about the lungs or the eyes being sterile, really nothing is sterile. Some locations are less densely colonized than others, and certainly our immune systems are effective at clearing microbes out. However, really nothing is sterile. Even, a mile under the Earth's surface or into the ocean, you'll still find microbes. And most microbes are not pathogens. It's really a rare minority of microbes that are able to cause disease in other animals. So most of the microbes that I've just been talking about are really something that we evolved together with, and they are not causing disease. Another thing that I would like you to remember is that every molecule can be used by a microbe. And so that means when you take medicines, even chemotherapy drugs, there are microbes out there that can metabolize those molecules. So that has big consequences for our diet and for our medicines. It has environmental consequences too, of course. So then looking at a human microbial community, you would expect to have maybe 10,000 different types of bacteria. As I've mentioned, the number of microbial genes greatly outnumbers the number of human genes. And if I were to take a ml of blood from any one person, I would find about 500,000 molecules in there. And we think that about half or so of them are produced or modified by microbes. So that means that all of the different molecules that are pulsing around your body and interacting with your different organs are affected by microbial metabolisms. Another angle that I would like to look at this from is where all that diversity is coming from, and so why it is that you're so unique from another person. And a big part of that is that there are phages that infect viruses, phages that infect each of the bacteria that colonize us. And there are continuous arms races between these bacteria and the viruses that infect them. And so each time a bacteria evolves resistance to a virus, it makes mutations in order to do that, and vice versa on the phages side. And so through the years, we evolve to have very diverse and unique microbial communities. And part of that is driven by the interaction between the bacteria and the viruses that infect them. So in my lab, we study the microbial communities in a number of ways. We use DNA sequencing to capture all of the microbial DNA out of clinical samples. And from there, we can learn about the taxonomy and the function of the different microbes that are present. We also use different mass spec approaches. So we can use gas chromatography and mass spec to capture the small volatile molecules that are present in these samples. And of course, our extraction approach will have a big impact on which molecules we catch. So, I mentioned earlier that there are 500,000 or so molecules in a blood sample. Well, each of these approaches will not catch all of those molecules. But if you keep things consistent, hopefully you'll be able to compare your samples through time. Similarly, we can use liquid chromatography and mass spec, and from that we capture larger, more polar molecules, again, depending on our extraction conditions. And so as a result, we can get a big picture of the metabolisms that are going on. And in many cases, we can understand the microbial contribution, although it's not always easy to tell whether a molecule has human or microbial origins. And then if we're lucky and we have an interesting hypothesis arise out of these data, we can test the hypothesis using in vitro culture models. And just to give an overview of what kinds of sequencing approaches we can take, we have a number of options for how to go about microbial sequencing. So for example, we can just look at one famous and well-studied taxonomic gene, the 16S ribosomal RNA. This is probably where 99% of the data in the microbial community field has been generated from. So in that case, you take your sample, store it, extract the DNA, and then you amplify up this one gene before doing the sequencing. So that's a little different than if you want to do whole genome sequencing, where you just isolate an individual microbe and sequence its genome. Certainly it's easier to assemble the genome that way. And then finally, I've also mentioned here in yellow that we can do metagenomic sequencing, where you take the sample and you really don't do much to it before you do the sequencing. And then you get shotgun metagenomes, which are in some ways more complex in terms of their analysis, but it's a nice way to get a big picture of all of the genes that are present in a particular environment. And then just to zoom in on the 16S RNA sequencing approach, because this is how 99% of the data has been generated in the microbiome field. So this is focusing on the bacterial 16S ribosomal RNA, which has conserved and variable regions that are shown in green and gray at the bottom of the slide. So the idea here is that we use PCR to amplify the 16S gene. We build primers in the conserved green areas, like where these red arrows are, and then we can amplify up that zone and compare it back to databases and use this information to determine the taxonomy of the microbes present in a particular environment. I would also like to point out that my favorite study design is longitudinal, because each person is so different from each other. If you do longitudinal sampling, then you can get samples from the same person and look at what changes through time and use their earlier time points as a baseline. So if you're following, for example, a cystic fibrosis patient, like I'll talk about later in the talk, if you catch them when they're feeling better, you have a good baseline for just them to compare to. If you do a cross-sectional study, it actually becomes hard to define even what healthy is in the first place. And so it's helpful to have longitudinal samples so that you can compare people to themselves. Okay, so today I'm going to talk about a few different projects, and we'll be able to answer a few different questions about them. So I'll first start talking about healthy humans, and then I'll tell you some more specific examples from preterm infant and cystic fibrosis patient projects. Okay, so here we are starting with the healthy humans. So first of all, I want to give some background on some of what we know about bacterial diversity in the gut around the world. So if you look in adult samples, well, actually, if you'll look at this plot, you'll see I have age going across the x-axis and the number of different types of bacteria going up across the y-axis. And if you look at adults in blue, you'll notice that Americans have less different types of bacteria compared to people who were sampled from Amerindia and Malawi. So if you look at age, you'll see that in the beginning of life, we start out with fewer bacteria, and then starting around somewhere between the ages of 3 and 5, the number of bacterial types stabilizes. And it stabilizes at a lower number for people from the United States than it does for people from these other two parts of the world. So that in itself is interesting, that it looks like there's less diversity in the guts of Western people. And now here's a plot summarizing an even larger number of data from a recent study that came out last year that was actually focused on the Hadza hunter-gatherers. And I've simplified the slide a little bit, and I'm showing you that here on the left-hand side of the plot, these are samples that came from people where they are less likely to have access to processed food. Whereas on the right part of the plot, you'll notice samples that come from parts of the world where people are very likely to have access to processed food. And the truth is they have very little in common. So our gut microbiomes have become quite different in the Western world compared to parts of the world where people have less access to processed food. And at the bottom here, you can see some of the specific microbes and their abundances in these different populations. So anyhow, that's pretty interesting. And there's a lot to learn about how the last 100 years of improved sanitation and vaccination and antibiotic exposure have affected our microbiomes. When I first started working in this field, it was about 10 years ago, and I was working in Geneva in Switzerland. And I did some projects focused on the healthy oral microbial communities by taking saliva samples. And so anyhow, this is a picture of Geneva. This was my bike ride to work every day. And it was really early before the Human Microbiome Project had started. And we did some simple studies where we collected samples from healthy people at multiple time points so that we could use the longitudinal approach that I talked about earlier. And some of the first parts of the project really were just about how to extract the DNA so that you could grab DNA out of very different cell types that had different types of cell walls. And then our goal was to analyze what kinds of bacteria were present and answer questions like how stable were the communities inside the same person. And so here's some results from that first project I was describing there, where the colors represent a particular person. So we have 3 time points from each person. And you'll notice in this tree that the people cluster with themselves. So that means that this is a way of answering the question, are the communities stable? Well, yes, in this group of people, a sample taken from the same person looks most like another sample taken from that same person. And it didn't matter exactly between the time points anyhow. So oral communities remain somewhat stable inside a particular person through time. And then we wanted to ask questions about whether that was true across families. And so in this project, we focused on families, and we took samples, 96 different saliva samples in this study from 4 different families that each had 4 people in them. So 2 parents and 2 children in each sample, in each family. And so we had 3 days in April, 3 days in June. And so that gave us some longitudinal information. And just to give you a sense of what was involved in constructing the project, we collected the saliva samples, we extracted the DNA in as unbiased a way as possible to crack open all the tough microbial cell types. And then we used the 16S PCR approach that I described earlier. We sequenced up those amplicons, and then we did some quality control to remove the bad quality sequence data. And then we used ecological statistics to ask questions about the alpha and beta diversity. So who was there and how different were the samples from one another? And I'll show you some of that data. So one thing that was certainly true that pops out immediately, even just looking at the data in a simple way, is that families share oral microbiomes. And so I'm showing you the data. There's one sample in each of the vertical lines here, and I'm clustering them by family. So you can see which microbial taxa were present in the different family members. Clearly, you can see that Streptococcus is a very abundant organism in all of the people in this study. But if you look at the relative abundance of some of these bacteria, you can see families 1 and 2 actually share a lot of features, as do families 3 and 4. And at the time that I did this study, TM7 in red, which is quite abundant in families 1 and 2, was actually not so well studied. And at first I was concerned that we had a contamination issue or something like that, although there is biology in that signal, because as you can tell, it's consistently present in families 1 and 2 and not in families 3 and 4. So if it was a contamination issue, you would expect a more widespread or random distribution of the TM7, which comprises 10 to 30% of the samples in some cases. So anyhow, that was an interesting discovery. And it's also interesting to think about the fact that the children were not that distinguishable from the adults, suggesting that kids are inheriting their microbiomes from their parents. And if their parents have a microbiome in their oral cavity that's associated with disease, then the kids are starting out with that starting point. So they're inheriting a potentially unhealthy microbiome from their parents. And so there's just a lot to learn on this front about what the impact of the microbial community colonization in the oral cavity is in different families, where some of them may be changing risk for particular diseases. So just to remind you of the strategy, we're doing a multivariate analysis of the community composition. In this slide, each of the colorful lines up here represents one piece of DNA from the 16S PCR amplification that I described. So we take each of these different pieces, we cluster them by sequence homology, where clusters 1, 2, and 3 could represent different species of bacteria. And then, as I mentioned before, we look at the samples and the different types of clusters This is an abundance matrix. It could be lions and tigers and bears in a different context, but here we're looking at different types of bacteria. From there, we make a distance matrix, and then we use various ordinations and projections, like looking at trees, like the one that I showed you before, to compare the samples to one another. So in one final representation of this family data, I want to show you how the families look compared to one another on this random forest multidimensional scaling plot. So on this plot, 2 points that are closer to each other have more similar microbial community compositions. And I've colored the families so that you can tell them apart. And so, for example, you'll see family 1 in red is more widely dispersed, but it's distinguishable from families 2, 3, and 4. And in the left-hand plot, I'm showing you the data at the phyla level. So it's, if you remember your biology, kings play chess on fine-grained sand, as in kingdom, phyla, order, and so forth. And so at the phyla level, which is a coarser level of detail, there's less ability to distinguish the families from one another. Whereas at the genus level, which is a more fine-scale level of information, you can separate the families from one another more clearly. And if you randomly gave me a sample from any of the families, with the phyla level information, I could correctly predict which family the sample came from 85% of the time. So I would have an error 15% of the time. Whereas at the genus level, it would be correct 90% of the time. So I can tell which family a person came from based on the oral microbes in their saliva. So that's kind of interesting in itself too. Okay, my next project that I want to tell you about comes from preterm infants. And I'll tell you a little bit about the gut microbiome composition in preterm infants at the Children's Hospital here in Orange County. And I'll also introduce you to an idea for a phage-related project, although I won't have time to go into detail with the data. So this is a study of premature infants that was conducted with samples collected here at the Children's Hospital in Orange County. It's a longitudinal sample, although it's retrospective, so it's not a perfectly designed study. You'll notice we have 77 fecal samples from 26 infants, and they had different health outcomes. Some developed necrotizing enterocolitis, some developed sepsis, and others remained healthy, although they were still being treated with many of the interventions that preterm infants are exposed to, including antibiotics. And we used 16S sequencing like I've described, and also gas chromatography mass spec to profile the metabolites. So here's one way of looking at the data that we got from 16S microbial community sequencing. You'll see we have the infants who had healthy outcomes, although they were stillborn preterm at the top. And then we have the infants who developed sepsis and those who developed necrotizing enterocolitis in the bottom panels B and C. This data has been published on the bioarchives, and where the paper actually was just accepted to MSphere, so you're welcome to look at the rest of the data in more detail. Really, the main thing that I would like to show out of this right now is that the most abundant bacteria in all of these samples, whether the infant had a healthy outcome or developed one of these complications, the most abundant bacteria is Enterococcus. And we actually could not distinguish the babies from these 3 different groups based on the bacteria that were present in their guts. Really, the main signal we're seeing is that if you're a preterm baby and you've been given a lot of antibiotics, most preterm infants go on a course of ampicillin and gentamicin. And so if you are a baby who's been exposed to antibiotics like that, your bacteria become very enriched for bugs like Enterococcus. And actually, Enterococcus also becomes enriched in adults who take antibiotics. So that means that a large number of babies are having the first months of their lives where their immune systems are undergoing really important development while they're being dominated by this bacteria and Enterococcus. Now, what we didn't see very much of is bifidobacteria, which is showing up here in pink. So a couple of the babies had a little bit of bifidobacteria, like number 17 here. And I think one of the other babies had a little bit of bifido. Maybe it was number 32. Oh, here we are, number 22. So bifidobacteria, from what we know in the past, actually dominated infant guts. And it's an important bacteria for digesting the energy in breast milk. And so here I'm showing you, this is also very fun to read about, so I encourage you to look into it. There are bacteria, bifidobacteria, that are able to access the energy that's found in breast milk. And in fact, there's even a company based on research that was done at UC Davis that's now selling this strain as a probiotic. And so we're still learning more about it, but it sounds like around the world and in human history, bifidobacteria was dominating the guts of babies. But that is certainly not what happens to preterm infants in this day and age. And actually, just to motivate our interest in Enterococcus, I mean, it's actually also an important pathogen in adults as well. It can become antibiotic resistant. So, now that we're starting to understand the importance of the microbiome in health, what can we do about it? 10 years ago, our main goal in our field was to understand what a healthy microbiome should look like. And I would say that question is still hard to answer, but we're starting to get there. But let's say we did exactly know what a perfect microbiome should look like. Well, how can we get there? What can we do to make that happen? So some of the options we have for manipulating the microbiome include taking probiotics and prebiotics. So for example, eating yogurt, which contains bacteria, eating fiber, which promotes the growth of healthy bacteria. Probably the most successful example of a microbiome research-related treatment is fecal transplants, which are often used to help treat people with Clostridia difficile infections. And then another option, which I think could be really powerful, is phage therapy. The idea of using viruses that infect bacteria as a therapy against bacteria. So why should we care about the viruses in the microbiome? Well, they're already shaping the composition and the abundance of the bacteria that are part of our microbiomes. But they also could be a potential therapy to treat antibiotic-resistant infections. And they've been studied in the lab in a few model systems like E. coli and Pseudomonas, but actually they haven't been studied that much using the kinds of bacteria that colonize our bodies. And as I mentioned earlier, these arms races between bacteria and the phages that infect them are something that generate diversity and are a big part of why each person has such a unique microbiome that's distinct from the person sitting next to them. This has been a big year in the United States for reconsidering the possibility of using phages as a treatment for bacterial infections. Now, phages were discovered in 1915 or 1917, depending on who you listen to. And they've been used as a treatment in the former Soviet republics. For example, in Tbilisi, Georgia, you have the Eliava Institute, where even today, people regularly use bacteriophages to treat bacterial infections. This box here I'm showing you has a cocktail of 6 phages in it. But there have actually been 3 cases, or maybe more by now, in the United States in the last year, where the FDA has given a compassionate use exemption, and Americans have been using phages here in the United States as treatment for resistant infections. So this example right here, I'm showing you, comes from UC San Diego, where a professor who developed an Acinetobacter infection was treated with the help of scientists from San Diego and Texas and the Navy, and he actually survived his infection. So his wife is Stephanie Strafty, and she's become a real proponent of the idea. And you'll see her out on Twitter encouraging phage scientists when there's somebody who needs a phage for a treatment. So this idea is gaining enthusiasm, but we also really need to study how it would work. And so that's part of what has motivated some of the projects that we've been doing in my lab that I'm not going to go into detail about, where we are starting to study phages that infect Enterococcus, like the ones that showed up in our babies. And so I'm very interested in the possibility of using phages to treat bacterial infections, and also just to shape the composition of the microbiome. So some conclusions from this part of the talk include that the health of the premature infant, as in their outcome of developing sepsis or necrotizing enterocolitis or having a healthy outcome, is not something that really drives the global composition of their microbiomes and metabolites. And our paper was just published, so if you're interested in that result, it's available. And then longitudinal sampling really is critical for relating the gut microbiota and metabolism data to health. And just that tools for shaping early life microbiota, for example, the probiotic bifidobacteria that I mentioned, and also the phage therapy, are on the horizon. And so I think these next 5 years are going to be really exciting for developing these ideas further. Okay, the final system that I would like to tell you about today is about the cystic fibrosis airways. And so cystic fibrosis is the most common inherited disorder among Caucasians. It affects about 1 in 3,000 people in the United States. And it results from a mutation in an ion transport channel that leads to thick, immovable mucus in the airways and chronic infections. So almost like having pneumonia all the time. And patients now have a median life expectancy of about 40. And that is largely due to the development of a huge diversity of antibiotics during the 20th century and beyond. And so you'll notice that, well, I can tell you in the 1950s and 60s that infants and children with cystic fibrosis were succumbing to infections from Staphylococcus and Streptococcus. And that is really not happening anymore, and people are making it into their 30s and 40s. Patients with cystic fibrosis now have children of their own, but of course with big consequences on their quality of life. But a big part of that has to do with antibiotics, pushing back the staph and the strep. And then now I would say that it's a different cast of characters in terms of the microbes that cause trouble in the airways of patients with cystic fibrosis. So the way I look at the kinds of microbes that are colonizing in the airways of cystic fibrosis patients is that they mainly group into 2 types of populations. And so you have bugs that are sometimes considered oral microbes. So many of them are anaerobes. Streptococcus is abundant, as you saw from our oral microbe data earlier, Rothia, Gamella, and so forth. And then in the lower parts of the airways, you have these long chronic infections with very slow growing and antibiotic resistant bugs. They're the ones that all these decades of antibiotic innovation have not been able to eradicate. And those include Pseudomonas and Stenotripomonas and so on. No matter where these bugs are having their main home base, where they're residing, they are producing molecules that are getting mixed up all the time. So if Streptococcus is producing fermentation products, those can actually get into the airways and impact the physiology of both host cells and also the gram negatives. So that's a bit of foreshadowing of some of the things that I've been finding. Here's an example of what the communities look like. At the top, I'm showing you the taxonomy from sequencing data, and at the bottom, I'm showing you the gene function from metagenome sequencing data. And you'll notice these characters I just have been telling you about. There's a lot of Rothia in this group of patients. Actually, there's not as much Pseudomonas, but you'll see some of the patients like this one here, the yellow patient, is dominated by Stenotripomonas, which is one of these slow-growing anaerobes, a gram-negative. You'll also notice the functions, which are more constant at this level of detail. And the truth is these remain somewhat stable for each patient. And really the holy grail in cystic fibrosis is understanding the periods where patient symptoms become much worse. So CF patients' lives are punctuated by periods where their symptoms become much more difficult, and they're referred to as exacerbations. They often have to go to the hospital and receive intravenous antibiotics in these times, but it's really hard to diagnose, and the microbial trigger is really not well understood. Some other foreshadowing are that there's lower pH in their sputum and breath samples, and we've been finding fermentation products from microbial activity, and that's what I'm going to tell you about. So our hypothesis was that microbial metabolites are good indicators of the CF disease state because we haven't found other ways to diagnose when a patient is about to get worse. So just sequencing the microbes has not really helped in the way that we were hoping 10 years ago when the sequencing first became possible. So people have somewhat stable microbial communities when you use the sequencing. However, it's not such a new idea. People have been using breath and even urine samples to profile the metabolites in human samples. So here's a paper from 1971 where Linus Pauling got a group of people, and he was thinking about the microbiome at that time, although we didn't have that word. And what he did was he asked people to eat a liquid diet with only small molecules, less than 100 Daltons in size, which essentially starved the gut microbiota. And as a result, he saw that their traces became more similar to each other. So patients on this simple diet had more similar breath and urine profiles using gas chromatography to trace them. Now we know more about what we expect to find in a human breath sample. Most of it is what's in the background air, but about 1% of the sample are these volatile organic compounds that I'll just refer to as breath metabolites in this study. And it's not always easy to tell if they have human or microbial origin, but these are the molecules that we're hoping to use to study health. And it's a very promising technique on both sides, really. I collaborated with Don Blake, and the Blake Lab here at UC Irvine has been studying environmental molecules and volatiles for a long time. In fact, their lab at the time, led by Sherry Rowland, are the ones who discovered chlorofluorocarbons and won the Nobel Prize in the 1990s. So they're a really high precision operation. And we worked with them to collect cystic fibrosis patient breath samples in these 2-liter metal canisters. One of the first things that popped out of this data is a microbial fermentation product called 2,3-butane-dione. And I'm showing you here 3 samples from a CF patient and 3 samples from a control patient. And you'll see that there's a lot more of this microbial fermentation product in the CF patient. So what is 2,3-butane-dione? Well, it's a pH-neutral fermentation product. It's produced in low pH and low oxygen conditions. If you brew beer, you might have heard of it because it's also called diacetyl, and so it can ruin the flavor of beer, and it can enhance other flavors as well. And it's got really interesting toxic properties that affect health. So for example, it's the main ingredient in microwave popcorn flavoring. And you may have heard that the people who work in microwave popcorn facilities, they have experienced bronchiolitis obliterans, and that is the result of exposure to this molecule, which is toxic to the epithelial cells lining the lungs. So here we are finding this molecule in the CF patients. And I'll show you a bit more data. If you look at the same patient through time, so these are days on the x-axis, and then this is the level of 2,3-butane-dione in the breath, you'll see the red samples this are relating to the CF patient. They're elevated in general compared to the healthy in gray or the room background in black, except for in this one time point. And in that time point, you'll see that there's actually not that much butane-dione present in the breath of the CF patient. And that's when the patient was taking IV antibiotics in response to an exacerbation. And so here's a signal that we can follow that, that changes with this patient's treatment. So we can't say whether it's an increase, a decrease in the production of the 2,3-butane-dione or an increase in the consumption. But our hypothesis here is that the antibiotics are blocking the microbe that's able to produce the 2,3-butane-dione. Furthermore, this level of butane-dione is enough to be destructive in itself. And so the red CF levels shown here are above the dashed black line, which is the safe exposure level dictated after the microwave popcorn factory disasters as a safe exposure level. So the molecule could be toxic in itself. We also have been taking sputum samples and analyzing them for molecules that are slightly less volatile. And here's an example of a patient coming in, and this is the moment of diagnosis of an exacerbation here at day 1. And you'll see they have much higher levels of 2,3-butane diol, which is a product of the same pathway, just a slightly less volatile molecule, a cousin of butadiene. You'll also see an amino acid degradation byproduct called putrescine, and aminovaleric acid, which is also a protein degradation molecule. So here's some other molecules we're finding that could be indicators of exacerbation. So I'm currently doing larger studies with more patients to see if these kind of signals remain true across larger numbers of patients. We also see some correlation between these increases in the fermentation products and amino acid degradation products that I'm showing here, and levels of Streptococcus, which is capable of fermentation, whereas Pseudomonas is not so good at that. So it could be that we'll be able to understand where the molecules are coming from. Okay, so I can see that I'm getting low on time here. I'm just gonna go down here. So we've been finding a number of different molecules that could be indicators of cystic fibrosis exacerbation. And each of these molecules has a reason for having its abundance change. And so each of them can help us get a window into what's going on in the metabolism and the health of the patient. So I think it's interesting that we've been able to put a GC on Mars or to detect these molecules, and yet we're still kind of in the infancy of studying the molecules in humans. I think there's a lot of potential for using metabolomics and other molecular detection mechanisms for studying health. In this case, with the 2,3-butadiene, we showed that we could detect this molecule, which tells us something about the environment. It's a pH-neutral fermentation product telling us that that this metabolism is happening in low oxygen conditions. We also could see that it's likely to be coming from Streptococcus and Raffia. We did some additional work to identify this out of our metagenomes, which you can also see in the papers that we've written about it. The molecule is directly toxic to human cells, and it can alter the physiology of neighboring microbes, such as Pseudomonas, which you can also read about in some of our follow-up papers. Furthermore, if you are interested in doing metabolomics-focused work, we've been working towards understanding how the samples need to be stored in order to be able to use the metabolomics data. So for example, if you take your samples and store them at 4 degrees C over the course of a couple of months, as I'm showing in this plot, you'll see that the composition of the metabolites diverges quite quickly within the first day, really. Compared to samples that were frozen at the -20. So getting your samples into the freezer the day that you collect them is pretty important for being able to have a stable metabolomic signature. And then just one final point that I would like to leave you with is comparing these two contemporaries, giants in the science of microbiology from more than 100 years ago. So Sergei Winogradsky, who's famous for developing Winogradsky columns that can study the layers of microbes in environmental samples, where the top layer will be using oxygen as the electron acceptor, and the next layers will be using the waste products from the layer above to carry out their respiration. He really focused on being able to study microbes in their natural communities, where you have a chance to watch their interactions with each other and with the environment. And that's just as true in the human body as it is in any environmental sample. Whereas Robert Koch, whose work clearly has been instrumental in expanding the human lifespan and discovering pathogens, he really focused on just isolating an individual microbe one at a time and studying it. And this is still the basis for a lot of the clinical microbiology that goes on. Many clinical samples are treated the same way that Robert Koch would have suggested in order to just study one microbe at a time. And so I think in these next 10 years, we're really going to want to put these ideas together and make sure that we're considering the impact of the neighboring microbes in the community on the physiology of the pathogens that we have been studying on purpose. Okay, so with that, I would like to acknowledge my lab members and many of my collaborators and the funding that we've received in order to carry out these projects. And with that, I'd be happy to take digital questions. And again, I'd like to thank you for the opportunity.